In this video, we will talk about plasmids and restriction enzymes and look at their history and use in gene cloning. The term plasmid was coined by Dr. Joshua Lederberg, who received a Nobel Prize in 1958 for his work on bacterial conjugation. Plasmids used in gene cloning are circular strands of DNA that were originally discovered in bacteria. Plasmids are found as independent DNA molecules separate from the bacterial chromosome, as can be seen in this image. The circular plasmid molecule is indicated on the right inside the bacterial cell. Plasmids have several unique properties that are useful for gene cloning. First, they are self-replicating molecules, as they can make multiple copies of themselves inside a bacterial cell. Second, they are non-essential for survival, meaning that the bacterial cell can survive even if it does not have any plasmids. Third, plasmids can integrate into the bacterial chromosome. And fourth, they contain special genes called antibiotic resistance genes that enable them to survive in the presence of antibiotics. In addition to their natural properties, plasmids have been modified to contain selectable markers, which will be described at a later time, and multiple restriction sites, which are sequences within the plasmid that allow the plasmid to be cut open and accept DNA fragments from other organisms, thus serving as carriers for these fragments. An example of a restriction site is indicated in the image on the right. The sequence GAA-TTC is an example of a restriction site. In order to understand the role of restriction sites better, let's look at enzymes that recognize and cut at these sites. These enzymes are called restriction enzymes. Restriction enzymes were discovered in the 1950s by Werner Arber. He won a Nobel Prize in 1978 for this discovery. They are molecular scissors that cut DNA at specific sites called restriction sites. On the right is an example of a restriction enzyme. This enzyme, called EcoR1, is bound to its target sequence within DNA. Restriction enzymes are naturally present in bacteria to guard against viral infection. Thus, they serve as a bacterial cell's immune system. So how does this immune system work? Bacteria are constantly under attack by viruses called bacteriophages. Bacteriophages attack by inserting their DNA into the bacterial host cell and taking over its machinery to make more copies of themselves. Restriction enzymes produced by the bacterial cell cut the foreign DNA, rendering it inactive. These enzymes recognize specific nucleotide sequences in DNA called restriction sites. Each restriction enzyme recognizes a unique restriction site. There are over 3,000 restriction enzymes discovered so far. On this slide is an example of a restriction enzyme called EcoR1, which recognizes the sequence GAA-TTC within DNA, binds to it, and makes a cut to produce staggering ends called sticky ends, which are complementary to each other. Now you must be wondering why the enzyme has such a strange name. Well, that's because the first 3 letters in its name are derived from the name of the bacterial species the enzyme was isolated from. which in this case is Escherichia coli. The remaining letters or Roman numerals indicate the specific strain number of the bacteria. Other examples of restriction enzymes include EcoR5, Hindi3, NDE1, BAMH1. The unique properties of plasmids and restriction enzymes make them useful tools for the process of gene cloning, which we'll look at in the next video. Thank you.